Hello, my name is Dominic Messina. I'm currently a chemical engineer going into my senior year here at Wayne State. My objective of this PowerPoint today is to present a summary of an article I found. Um, I found it through Scientific American. It was entitled "New Thalidomide-like Therapy Hijacks Cell Trash Disposal System." This was uh, this was posted by Karen Weintraub on May 23rd of 2018. So as we know, summaries will be used quite a bit in the workplace, especially for engineers, as we'll be getting quite a few technical documents, and the main points of these documents will definitely need to be shared with your peers inside of your team, with management, and even with workers down on the floor of a production plant. Very quickly, I'd like to just give a forecast of what I'll be talking about for the rest of this presentation. Basically, I'll just be going over the main points of Weintraub's article, those being the mechanism of action, or how the drugs interact with other molecules in the cell, the impact of this technology on the pharmaceutical and medical industries, and then the future of protein degradation therapy. And then finally, I'll end with conclusion and the main points of the article. So basically, Weintraub kind of started out by giving a little bit of history on a drug known as thalidomide, which we know can treat cancer, but was actually a little bit more famous for causing birth defects back in the 1950s and 1960s, where it was used as a morning sickness pill. And for a while, scientists were kind of confused as to why this happened. It turns out it has the same mechanism inside the cell. Basically, thalidomide would go up, attach itself to a protein, and kind of tell the cell to get rid of this protein, get it out of the cell. Which is good when you're trying to remove toxins from, say, like a cancer cell, but in the case of thalidomide, it was actually removing a, a vital protein from which was needed for fetal development, which was causing some pretty severe birth defects. But basically, this research is implemented to what is known as protein degradation therapy, where they're starting to develop pretty well-engineered drugs to attack specific proteins, such as the amyloid beta protein, which builds up in a Alzheimer's patient. Um, Potential side effect is this therapy could attack more than one protein. So like in the case of thalidomide, it can do one good thing and then have a pretty severe side effect. So Weintraub kind of goes on to talk about how the mechanism of action differs pretty fundamentally from how we're used to doing pharmaceuticals these days, or at least how we attack diseases such as Alzheimer's. Basically, we use what are known as small molecule drugs, which will attach itself to a active site on a protein, thereby deactivating it. Whereas in protein degradation, it just removes the entire protein. And we're seeing a push from pharmaceutical companies and a lot of biotech startups. They kind of realize the potential of these drugs, especially in their ability to easily modify them to work on a range of specific targets. So even though this technology is pretty promising, it's actually mostly untested on humans. Probably the most cutting edge study to this day was done in 2015 on some lab mice, where they created a pretty specific drug aimed at degrading the BRD4 protein, which is essential for leukemia growth in both mice and people. And as I was saying, we're actually able to get more and more specific with these drugs now to where they'll only attack one protein instead of like multiple, where we can have a number of side effects. And actually, the most useful pragmatic use of this technology to this day would be in documenting the use of the 20,000 plus proteins in the human body. So they're going through one by one, deactivating a protein and seeing how the body responds to it. And lastly, it was noted that drug production is outpacing the knowledge of the mechanisms at this point. The example that Weintraub used is our ancestors using willow bark as a as a painkiller when we know now that it's actually a precursor to acetaminophen. So lastly, I'd just like to conclude with a couple of main points from the article, namely Weintraub's desire to inform the reader of what is known as protein degradation therapy, kind of its history, where it came from, how we're moving forward with it today, and the potential future uses. Obviously describing the impact of this technology on the pharmaceutical and medical industries, and kind of how these industries are adapting to new technology. Obviously, as I said, the relevant uses today, and foreshadowing what we can do with it in the future. And lastly, the importance of just spreading knowledge, especially stuff you believe in, especially cutting edge technology, so we can kind of get more people involved with this, kind of speed up the process a little bit, and get it going to the general public as soon as possible. So that'll conclude my presentation. I hope you enjoy. Have a good day.